Hello, everybody. My name is Minhua Liu. I'm the director of the Center for Translation and a professor in the Department of Translation, Interpreting, and Intercultural Studies of Hong Kong Baptist University. On behalf of the arts faculty and the Center for Translation, I would like to welcome you all to today's session of Arts Does Method. Arts Does Method is a series of lectures and workshops provided by the arts faculty to focus on the methodology of doing research and are catered specifically to research postgraduate students and young scholars. And today's session will focus on the neuroscientific approaches to translation and interpreting. And there's probably no better speaker than Dr. Adolfo Garcia to talk about today's topic. For those of you who attended our international conference on interpreting last month, and I'm sure there are quite a few among our participants today, you have already met Dr. Garcia and have heard his fascinating talk, where he gave a comprehensive overview of how bilinguals and specifically interpreters process information in their brain. In his talk today, he will get into the nitty-gritty of studying translation and interpreting from the perspective of neuroscience. And I'm not going to say more about Dr. Garcia's background, as we did, because we did plan to leave a lot of time for questions. But I wonder, Adolfo, maybe you can also tell us a little bit about your career trajectory, particularly how you became a scientist, from a translator and an English teacher. So the floor is yours.
Thank you, Minhua, and good morning, afternoon, evening, night, weeny hours of the day, wherever you are. Thanks for joining us here. It's a pleasure to have this opportunity to share ideas with all of you. And before I answer Minhua's question, let me just say that this is not going to be like an in-depth methodological workshop into any of the approaches that we're going to be looking at, just because time would not allow for that to happen. Instead of doing that, what I would like to provide you with is an overview of the different methods that brain sciences at large provide us with in order to explore translation and interpreting processes from a neurobiologically grounded perspective. So my idea today is to show the main properties, the main contributions and limitations of different approaches. And then those of you who may be interested in delving deeper into any of those, there will surely be opportunities for us to do that. Now, as to Minhua's question, something that perhaps is worth mentioning is that I come from a humanities background. I hold degrees in translation and foreign language teaching, and I was mainly trained as a linguist in the early years of my training. And at some point, I was recruited by an interdisciplinary group that was building up in New York at New York University. And this was a group of neuroscientists and neuropsychologists who wanted to explore language from a neurobiological perspective. And they wanted a linguist who had a background in bilingualism and translation, because they dealt with lots of bilingual patients. So they had read some of my earlier work, and apparently that did not deter them from contacting me. Which is something that to this day it doesn't cease to amaze me. And I remember being there and being invited to join a direct electrical stimulation session with a patient. And what they were doing there was they were providing direct electrical shocks directly onto the brain of this person as he was awake. It was an elderly person here, and I was absolutely fascinated to see that by applying an electrical shock in certain brain regions, this person would be unable to name pictures in one of his two languages while remaining fully capable of doing so in the other. And that was a sort of a life turning moment for me. And it was at that point that I said, okay, if I want to study translation and languages in general in a way that I feel comfortable with, and I've always had this sort of uh, proclivity towards biological aspects of life, then I better start getting serious about neurolinguistics. And that was basically my transition point. I struggled a lot in that complementation of my humanities background, moving on to the world of statistics and neurophysiology and modeling and things like that. And it was and it remains to be a very, very enjoyable ride. And one of the things that makes it more enjoyable is to learn about the methods in particular, and to see that there are so many ways in which we can spy the brain, which we can peep into the brain and gain insights, always indirect and always partial, but at the end of the day, as useful and as informative as the insights and observations that we can derive from any of the other approaches that have become traditionally mainstream in translation and interpreting studies. So I hope that this suffices as a little bit of an introduction, and I'd like to share my screen now and start sharing with you some of the things that I prepared for today. As I'm doing this, I will not be able to take a look at the chat window, and we do have a substantial amount of time allotted at the end of the talk for questions. That being said, if anybody wants to interrupt me or make a comment or raise a question as I am talking, please do so. And I think that the best way to do so would be if you just unmuted yourself. I wonder if that's possible, and did it. And if not, maybe you can just drop a comment on the chat window, and maybe Minhua or Esther, they can read out that comment for everybody. All right. So let's get started.
Yeah, Rodolfo, that is probably during your talk. Probably it's not that easy for us to allow people to unmute themselves. So
All
probably
right.
Yes, I agree that probably the better way is to type in your questions in chat, but we may not entertain your questions immediately during the talk, unless you have we see that you have an immediate question about a specific point, and that probably that can be asked at the answer at that time.
Fantastic. So let's do it that way then. Thank you. All right. So it's a treat to be here with you at the Art Does Method series, and we're going to be talking about translation and interpreting. And if we take a little bit of a historical view here, I'd like to take about what I would like to what I like to refer to as a constant in a constant. So, the need to broker between languages has been a constant of humanity, right? Ever since two speech communities first came into contact to exchange goods or discuss how they were going to divide a territory or to engage in more complex socio-cultural or political negotiations, the need arose for people to exchange messages in more than one language. And think, about, for example, Saint Jerome working by candlelight, or a dragoman serving the Ottoman Empire, or a religious translator in the Renaissance, or maybe even a Jesuit missionary operating in China, during the Qing dynasty, or a conference interpreter at the Nuremberg trials, or if you want to take an even more contemporary approach, think about any of your translator colleagues working at home on Trado Studio for an agency that's based elsewhere in the world. So certainly, across this evolving narrative of translation and interpreting, there have been countless changes. Just by taking a look at those pictures, the topics that required interlingual brokering, the type of training or lack thereof, formal training that practitioners had, the technological devices that were available, the very socio-cultural settings in which interlingual mediation took place. All of these things have changed and continue to change to this day. But there is one thing that has remained unaltered across this centuries-long history, and that is the fact that human translation, as we know it, has always relied on particular patterns of neurocognitive activity. And for different reasons, some of which we may try to sketch out today, the role of the brain in translation and interpreting has typically fallen beyond the radar of translation and interpreting studies. And one may ask why this is the case. And I think that there are a number of reasons, and I will not try to provide an exhaustive list, but just to mention a few of them that I think are worth mentioning. First of all, translation and interpreting studies have long been rooted in a very prolific, informative, thought-provoking, exciting humanistic tradition, employing tools and profiting from insights from linguistics and philosophy and social studies and gender studies and post-colonial studies. We've learned so much about the way in which translation and interpreting are deployed in a myriad of settings, just by using the toolkit that humanistic disciplines provide us with. The second is that the foundational evidence that actually had something to say about the role of the brain in translation and interpreting appeared in somewhat obscure and mostly inaccessible journals. Indeed, evidence about the role of or how brain lesions could impact different translation phenomena has been available, at the very least, to my understanding, since 1929. And still, if you take a look, that evidence was very, very largely neglected until very, very recently in the field. And this is probably more because of the lack of access, of widespread access to those findings than because of any genuine lack of interest on it. We should also mention the relative newness of neuroscientific methods. Translation and interpreting studies, although there's some debate as to when the discipline could be said to, my apologies, could be said to have its actual birth date. It's been, reflections on translation and interpreting have accompanied us for, that's my phone, my apologies. Have been made for decades and decades on. Now, it's only recently that neuroscientific tools have become widely available. If you think, for example, about the very, very first neuroscientific or neuroimaging studies on linguistic processing, for example, word reading, they are not that old. They date from the very, very late 1980s. So again, and there's also this relative novelty of the very emergence of cognitive neuroscience as another line of insights into translation and interpreting. And then, of course, those tools have been applied to study translation and interpreting even more recently. Now, what I would say too is that I think that the time has come for a change, and this is not just a wishful enunciation. There are specific changes that have taken place that have enabled neuroscientific perspectives to actually fall within the mainstream radar of translation and interpreting studies. And this is the case because of some developments that have enabled these this change, let's say. First, in the last decades, translation and interpreting studies has fully embraced experimental approaches, ranging from insights from psycholinguistics and cognitive science, mainly, and as well as experimental psychology. Nowadays, drawing on experiments, which are, by the way, the backbone of cognitive neuroscience, is not something that is perceived as odd in some of the mainstream strands of translation and interpreting. Then neuroscientific literature in general has become much more widespread, accessible to everybody. So it's no longer a challenge to go and dig up relevant findings regarding any cognitive process that may be of interest to readers or scholars. Neuroscientific tools have become much more popularized than they were maybe 2 or 3 decades ago. Not only is it more frequent to find MRI scanners or EEG equipment in different universities and research settings across the globe, but they are no longer the exclusive property of let's say, psychology departments or biology departments. These tools have become much more widely available. We even have laboratories across the world right now that have these tools and are directly and specifically devoted to studying translation and interpreting. So this is also very good news, I would say. And there's another thing that has to do with the communication across scholarly traditions. On the one hand, there has been increased attention to brain-related aspects from the translation and interpreting studies community. And that has been echoed by an increased interest in translation and interpreting on the part of cognitive neuroscientists. So we have this literature in which there are at least 2 strands of research that not always come together, I have to say, but there are 2 different ways and synergistic ways in which we are learning more day after day, maybe week after week, with new publications about the role that different neural mechanisms play in intralingual reformulation in all of its guises. And there are dozens of relevant studies, and that number keeps increasing. And that's also very good news. Now, one could still ask oneself, what's to be gained by this? Because the truth is that we have been able to infer cognitive determinants, aspects, characteristics of translation and interpreting in multiple ways for decades without any need whatsoever of looking at the brain. So going back to the late 1950s and the early 1960s, we started gaining insights and of whatever quality you may deem them into cognitive aspects of translation by extrapolating insights from rationalist linguistic models, such as Chomsky's foundational transformational grammar schemes. Also, around the same time, but not so much in the US as in Europe, another trend emerged which pretty much advocated for engaged in active observation of practitioners, mainly simultaneous interpreters at work. And these observations, together with um, the analysis of note-taking, for example, in different interpreting settings, also led to quite popular and to this day present cognitive accounts of interpreting and translation proper. And again, no mention of the brain was needed for this to happen. Jumping forward a little bit in time, think aloud protocols were also instrumental for us to learn about cognitive effort and to have our first empirically grounded insights on translation strategies and errors and problems. And this is a technique to this day remains very useful. But other than introspection, we have also developed ways of looking at text products, translation products, actually written texts in corpus studies. And again, different automated and theoretical tools have been developed to derive cognitive interpretations of patterns that could be gleaned from written texts, translated texts, that is. Think about eye tracking, or think about also other behavioral measures, such as measures of accuracy and response time in more classical psycholinguistic studies. Those have also been used for decades now to learn about cognitive effort, distribution of attentional resources, or even to model the strength of the links connecting different words in the bilingual mind, if you will. So this is a very diverse, rich, and useful toolkit that has accompanied the field for decades. And this has prompted specific hypotheses, models, even this has inspired specific approaches to translation training, and many, many other things. And of course, the brain was completely unnecessary in all of those pursuits. Now, if we have achieved all this in the history of translation and interpreting studies, and the brain was absolutely unmentioned in all of them, then again, what's to be gained by looking at the brain? And something that I should say is that for one avenue of insight, not to be indispensable for the development of a field, does not mean that that line of insight is superfluous. So allow me to substantiate this point a little bit. A very basic thing that we could say is that the mind and the brain are consubstantiated, and that they hold predictable relations. The specific neural regions and networks, the specific neural dynamics that underlie the different cognitive, affective, motoric, interoceptive, mnesic processes, they are not randomly related or distributed across neural regions and mechanisms. On the contrary, although there is enormous inter-individual variability, there are very consistent patterns. And if you're interested in learning something about the mind, then certainly looking at the main organ whose integrity is crucial for the normal functioning of the mind is something far from an eccentricity, I should say. What specifically can we learn by leveraging neuroscientific tools? And this is where we reach the actual methods part of the talk. Something that we can learn about is the functional organization of relevant systems. So there are basically, you can ask yourself, and this is a very legitimate and interesting question for someone who's interested in cognitive aspects of translation or interpreting, it is to say, okay, so I can be quite sure that when I am translating or interpreting, many, many different cognitive systems are coming together. So I know that the activity of translation taxes my lexico-semantic systems. It requires my attention. It involves work organized. How many of those systems can be dissociated? How many of them can actually be identified as partially autonomous components? How do they, does the level of performance that I achieve in one of them necessarily determine the level of performance that I can achieve in any other one of them? And these kinds of questions can be partially, of course, but still robustly approached from a neuroscientific perspective. And there are two main methods, or two main, I should say, perspectives in brain science that are useful to this end. One of them involves lesion models, and the other one involves functional neuroimaging. Regarding lesion models, this is basically the foundational, the cornerstone, if you will, of experimental neuropsychology. And he had to profit from this approach to learn about aspects of translation across decades. Let me tell you a little bit about this. Basically, at its core, what this approach involves is working with patients who have sustained brain damage. The literature has mainly favored patients with focal brain damage, mainly due to a stroke, which could be ischemic or hemorrhagic. And usually what happens in stroke is that will produce a very focal and typically stable lesion. That is to say, unlike what happens in neurodegenerative disorders, such as Alzheimer's disease or Parkinson's disease or behavioral variant frontotemporal dementia, where you have actually progressive atrophy that will typically start in a core area and then progressively expand and degenerate tissue across different brain areas. What happens in focal lesions is something that's quite the opposite. I mean, the lesion will be very focal and will remain focal for a certain amount of time, depending on the cause, the etiology of that lesion. But basically, you can study a person that has that focal brain lesion, let's say a lesion that has compromised aspects of the inferior frontal gyrus, and you can administer a set of tests to that person. And usually what you want to do is you want to include contrastive tests, tests that allow that share some property but that differ in a critical property. So, for example, you could test a person in tasks in which you assess their syntactic abilities during linguistic production versus their syntactic abilities when they are listening or reading, listening to language or reading language. You could explore maybe dissociations between syntactic abilities and lexical-semantic abilities. Or, as has been done in the field, in bilingual patients, in bilingual persons with aphasia, you can, for example, compare the level of performance that these people with lesions in circumscribed brain areas have when they are translating, for example, certain source units, let's say words versus sentences as source units. Or you can compare their skills when they are translating in one direction, let's say from their second language into their first language, than when they are doing it the other way around. And this is something that has indeed been done, fortunately, in a number of studies across the years. And something that we've learned this way is that, first of all, there are different ways in which translation skills may become dysfunctional upon sustaining a brain lesion. Some people manifest compulsive translation behavior. That is to say, after sustaining their brain lesion, they find it impossible to suppress the tendency to translate into other languages what they have just said, or what their interlocutors are telling them, or even what they hear in the environment from third parties. Another translation neuropathology that has been discovered is called inability to translate. And this is basically an incapacity to engage in translation, either in both directions or even in just one direction, even when your single language production and reception skills, let's say your capacity to produce and comprehend language only in L1 or your capacity to do so only in L2, are partially or fully spared. There are two other forms of translation disruptions that have been documented. We can talk about them later. But if you take a look at what I have here, it's a selection of a number of case studies in which participants manifested inability to translate. And what you can see here is that there are, depending on lesion site here, so which hemisphere and which particular area was affected, you can find different patterns of affected or preserved performance in different translation directions. And this is one of the ways in which brain science allows to establish brain behavior correlations. This way is particularly called anatomo-clinical mappings or anatomo-clinical correspondences. Basically, if you find that a person has a lesion in area A, and that person is able to perform task Y, but not task Z, but at the same time, you have another patient with a lesion to area B, and that person is unable to perform task Y, but that person is able to perform task Z, you have what is known as a double dissociation. And that is substantially strong evidence for you to propose that the area that was observed as compromised in the first patient has a particular relation to the function that is affecting that patient. And by the same token, that the area that was affecting the second patient does play a particular role in the function that was affecting that patient. And this is, as I said, a very a fundamental line of evidence for cognitive neuroscience at large, even to this day, is very good that we have been able to incorporate this into our understanding of the translating and interpreting brain. And you know, when I say a good thing, there's always this sort of paradox, because what this approach does is to turn the tragedy of another person into an asset for a scientific field. These patients, they are in a way unwilling martyrs of cognitive neuroscience. These natural accidents that happen are tremendous for the patients. They can disparage their personal lives and that of their families, but at the same time, they provide such a rich body of evidence that we will always owe a great debt of gratitude to the time and the effort that the patients devote to these studies, even after they have endured this this episode in their lives. Just for you to see one specific example, this is a very well documented single case study in which a bilingual patient who was a native speaker of Friulen, an Italian dialect, and then who had learned classical Italian during her education, upon sustaining a brain lesion that mainly compromised frontal-basal structures, particularly the basal ganglia. If you take a look at her performance in backward translation, this is from L2 to L1, and forward translation, this is from L1 to L2, you will see that systematically across different translation units, words in an oral modality, words in a written modality, and sentences in an oral modality, her performance was better when she was translating from L1 to L2 into L2 than when she was doing it the other way around. So this is very interesting because this suggests that area, in particular the basal ganglia and the frontal basal networks that are associated with it, might play a more crucial role in backward translation, in going from L2 into L1 than in the other translation direction. And this is one of the ways in which we can start to find possible candidate neural correlates of particular translation subskills. But I said that there's another approach by which you can raise and answer questions about the functional organization of translation and interpreting systems, and that's by relying on neuroimaging evidence. There are different neuroimaging tools. Here, I'm just going to make a very, very quick reference to functional magnetic resonance imaging, for short, fMRI. You may be familiar with this tool, which basically yields images, brain images like the ones that you are seeing here. But what's interesting is reflecting a little bit on what's happening here. And it goes, the story, if I have to make it very, very simplistic, goes like this. Whenever you're engaging in any cognitive activity, when you are trying to remember to evoke the image of an actor, the face of an actor in your head, if you try to do it now, when you're singing or humming a melody to yourself in your mind, when you are looking at a change in a light pattern in the room, when you're listening to a word, when you're comprehending a sentence, when you are feeling aroused, excited, sad, when you're moving your toes, anything that you're doing will involve specific patterns of brain activity, and those, and in particular, brain areas. And those brain areas that are more critically recruited for the experiences and the tasks at hand, they will have greater metabolic demands. In order to do what they are supposed to do, those brain regions will need more food, if you will. They will need more fuel. And that fuel will basically reach those regions through the bloodstream. And what happens basically is that you will have hemodynamic flows reaching that area, and the cells that are involved in the function that needs to be deployed, they will extract the oxygen from the red blood cells that are reaching that area. And when you remove oxygen from this this way, what you have is you go from having oxyhemoglobin to having deoxyhemoglobin. And the deoxyhemoglobin, that's the hemoglobin without oxygen, has paramagnetic properties. And those paramagnetic properties can be captured with a gigantic scanner like this. So when you have a person lying there and you have them engage in a linguistic task, what you will see is that different brain areas will have greater metabolic demands, they will consume more oxygen, and that sort of paramagnetic residue can be captured with the scanner. And that's basically what you see. Yeah. So this is a very indirect way of detecting which brain area across the whole brain is actually consuming more resources for the task at hand. And you can visualize that by then plotting colors on a template of a brain. And that's what we see. Now, this technique that has very, very good spatial resolution. That is to say, it's a very good technique for you to answer questions of the type where, which part of the brain. So if you have more of a regional or localization type question, then you're going to be doing very well with this technique, if you design your experiment properly, of course. But this technique at the same time is very bad if you want to answer temporally specific questions. Another way to say this is that this technique has very poor temporal resolution. Usually, from the moment in which the process at hand begins to be deployed to the moment where that paramagnetic residue can be captured, as many as 1 or 2 seconds can actually elapse. And the sluggish nature of the bloodstream in the brain makes this technique very bad to answer temporally precise questions. So, and 1 second, 2 seconds in time in terms of neurocognitive activity is just too much. It's an entire lifetime in a way. So that is why it's good to be aware of both the pros and the cons, the virtues and the limitations of each technique. And this technique has been used to explore translation interpreting processes in different studies, and especially in recent years, which is quite encouraging. There's this one. This is a classical study that actually is interesting because it's one of those pioneering collaborations between an expert in neuroscience, such as Dr. Rina here, and an expert translation scholar and translation interpreting scholar, such as Jorma Tomola here. And what they did here was to compare brain activity patterns as bilingual individuals engaged in interpreting tasks in both L2, L1 direction and L1, L2 direction, and also when they engaged in shadowing, which most of you I'm sure will know, so excuse me if I say something that's obvious, but when we say shadowing, we mean a task in which you listen to unfolding discourse in one language, and with a minimal gap in time, you repeat what's being said in the same language. So in a way, shadowing represents a sort of single language or monolingual version of interpreting, if you will. And it's a good control condition in this way because you have basically many of the same cognitive control mechanisms involved in the task, but without the added load of changing between languages for reception and production. And what was observed here was that, first of all, there was a lot of overlap in the brain areas that participated critically in L2, L1 interpreting relative to L1, L2 interpreting. But at the same time, another thing that was observed was that those rather overlapping activity patterns were actually wider and of greater magnitude in L1, L2 interpreting than in L2, L1 interpreting. And this adds to a body of evidence that shows that typically when you are translating from your strongest to your weaker language, you usually have more widespread activity patterns, which have traditionally been interpreted as a neurobiological signature of greater cognitive effort. But there are more precise things that we can say about it, and we are going to reach that point shortly. But basically, what I want to show you here is that you can leverage neuroscientific tools to also study the functional organization's translation systems in real time. And here's the main difference. When you are using lesion models, basically you are not observing brain activity as it happens. You may know that the patient has a lesion in a certain brain area because that person has received a brain scan before the test, and that's perfectly good and it's tremendously useful. But you're not really seeing what's happening in the brain of that person as he or she is performing the task of interest. That is what you can do with this. This is in vivo evidence, and it's a way to really peep into the brain as it is engaging in the process of interest. So it's very interesting that we have now the opportunity to combine different strands of evidence towards more multidimensional and comprehensive views of, in this case, the functional organization of translation systems in the human brain. But there are other things that we can do. Another thing that you may be interested in exploring is the inner unfolding of the intervening processes. So I told you, these two approaches, they are fantastic, but they are not really good if you want to know when something is happening in the brain. That's fortunately a type of question that we can answer by using specific neuroscientific techniques. In particular, there are two techniques that have been used in the field. And the first one is electroencephalography, or EEG. So let me tell you a little bit about this method. That, by the way, is my wife that you can see on the left of the screen. And she happens to be a professional translator. So oftentimes, not so much these days, I have to say, but back in the day, whenever we were designing new translation experiments, we would have her come to the lab and we used her to pilot the experiments. So she has done her share of contribution to the research that we've been conducting in this area. But basically, what I want to show you here is how this is set up. You first attach a series of electrodes, and there are different devices. Some of them have very few electrodes, 32. Some others have 64. We work with high-density arrays, which have as many as 128 electrodes. So it gives you a more comprehensive scalp coverage. And what you can see here on the second picture towards the right is the main experimental setup. So the participant, once all these electrodes have been applied on their scalp, they will be sitting and they will face a computer, and different stimuli will be presented there. So they're gonna be performing a task. So here, what we do is we present the translation tasks. Maybe we'll show them words in one language and the other, and they will be asked to either translate those languages into the other language or to read them to have a baseline for comparison. And as the persons are performing the task, we will be recording their brain activity through the electrodes. And you can see now all those cables, they're going to be sending electrical signals through that robotic arm onto a separate room in the lab, where we're going to be observing in real time, millisecond after millisecond, the electrical modulations of the brain as it is engaging in the task at hand. That's what you can see in the bottom figure here. And this is a very, perhaps the most classical technique in cognitive neuroscience. And from those electrical signals, you can derive many things. You can do many things. The first thing I want to say is that this technique has very good temporal resolution, right? Remember, I just said that you can measure these changes millisecond after millisecond, and that is a level of temporal precision that is just the gold standard in the field right now. However, this technique is not very good to answer questions of the type, where are things happening? You'll see these electrodes, they are placed on the scalp. And you would say, well, if one electrode is the one that's mainly leading the modulation that I'm observing, then certainly that brain area below the electrode must be responsible for it. And that will be wrong. Because these electrodes, they are sensitive to electrical signals coming from a very widespread surface, with estimations suggesting that it may be as many as 10 centimeters around the main point of the electrode. And at the same time, maybe an electrical peak that you observe in a certain electrode may even have generator sources in subcortical areas or in counter-hemispheric areas. So really, it's very hard for one to derive any regionally precise interpretation from this. So, there's a simplistic way of presenting the tandem of fMRI and EEG, which is a little bit of a simplification, but it's really not that off. So just like I said that fMRI has good spatial resolution and bad temporal resolution, you can very firmly say that EEG is characterized by very good temporal resolution in the absence of good spatial resolution. Maybe this, I don't know if all of you have had a chance to perform an EEG at some point in their lives, but most certainly you have at some point done an ECG study, electrocardiogram, and you will have some electrodes placed around your chest, around particular areas, the heart area, and you will have a millisecond by millisecond register of peaks and valleys that are printed on a page or that appear on a digital format, and it's very much the same logic. And the key point here is that those changes in electrical pattern are not random. When there's a certain negative peak in voltage at a certain point in time, you can derive a specific interpretation of that, because that specific modulation does not happen just because. That in particular may be indicative of how effortful it is for you to integrate a word into the conceptual scheme that the preceding sentence offered. And this is, again, a highly complementary and highly useful approach to study the brain with a temporally precise way. Now, there are many things you can do with this. You can study event-related potentials. So you can see exactly what I was just mentioning, at which point in time there's a peak or a value that is significantly higher or lower in amplitude than another condition. And you can say, okay, so at this point, I have this modulation, and you can interpret that in a specific way. That's event-related potentials. And you may be familiar with this. Some of them have become very well known, such as the N400, the P300, the N2, the P600. All of them are neural traces or indices of particular types of processes that the brain is engaging in at some particular point in time. But you can also do other things. You can focus on how electrical signals are being modulated in specific frequency bands in time. And this is something that we are using in this work that we are finishing right now, in which we explored oscillatory correlates of forward translation, again, from L1 into L2, versus backward translation from L2 into L1. And we are trying to see whether the way in which different neural networks become synchronized and desynchronized in time vary among these translation directions, and how they unfold in different time windows. And something interesting is that as you move through time, the specific frequency bands that exhibit significantly different patterns of synchronization or desynchronization between one translation direction and the other are different in an early time window. For example, in the first figure here, you can see a pattern of greater neural synchrony in this frontal or bilateral frontal cluster, in a range between 5 and 7 hertz. But then, if you move forward in time and you see what happens from 300 to 600 milliseconds after a person is translating a word, what you will see is that forward translation is characterized not by greater synchrony, as was the case earlier, but by greater desynchronization than backward translation, especially over a higher frequency band. That's the higher beta band. So this is very interesting to understand that not only do different translation processes have specific temporal dynamics, but that those temporal dynamics can change both in terms of the level of synchronization coupling or desynchronization decoupling of different neural resources, and also in which frequency band along the many bands along which the brain oscillates are indicating those changes. And I said that there's another way of looking at temporally precise data, And this is by using intracranial EEG. Like I mentioned, in what we just saw, we have signals that are being captured on the surface of the skull, right? So you place the electrode here on your skull, and of course, between the electrode and what you're actually interested in capturing, which is the electrical signal, there's all sorts of sources of noise. There's hair, there's skin, there's bone, there are all sorts of substances in between. So what you have is a very degraded and impure signal. And of course, it's coming from, you can't really tell where the signal is coming from. What you have is more of a broad representation of the overall activity, the overall changes in electrical activity, either across the whole scalp or in certain clusters that may be of interest. Now, there are very special circumstances in which you can have all the highly precise temporal resolution of EEG in combination with an optimally precise spatial resolution. And that is the unique combination that you obtain when you use intracranial recordings. There's a proportion of patients around the world with epilepsy have what is known as refractory epilepsy. So, and these patients, the vast majority of patients, you can treat their disorder with pharmacological treatment. But there's somewhere between 10 and 6% of patients who will not really respond well to pharmacological intervention. And in those cases, the only chance that you have to actually treat their disorder, especially if it's severe, is to detect which brain area is responsible for triggering the epileptic seizures so that you can then remove it, or at least provide the patient with an informed consent and information so that the patient can know whether he or she wants to undergo such a procedure. The point is that, the doctor will tell him, we have detected which part of the brain is responsible for your seizures, we can remove it. But if we do, and that would probably put an end to your seizures. However, we have to tell you that this brain area is crucial for your linguistic abilities, or it's fundamental for you to move your right arm and hand, so that there's a chance that when we remove it and we put an end to your seizures, you may lose this or that function. So that's basically why these procedures are done. And the way in which this works is that very, very, very small microelectrodes are implanted directly inside the brain of the participant. And each of those, it's like little spears, right? Like little needles. And each of those needles will have a number of electrodes in them. And they will be implanted directly inside the neurons of the person in different areas. And those electrodes will then be inside different aspects of the brain. And this is one very direct way of detecting. So you're going to do this, and the patient is going to be in the hospital or the clinic for maybe a few days without doing anything in particular. Basically, doctors are going to be waiting for that person to have a seizure. And when that person has a seizure, you can go back and see the recordings from the electrodes that you had implanted in the person and detect where those seizures originated. So this is done basically with pre-surgical, for pre-surgical reasons. Now, during those 2, 3, or 4 days that the patient has all these electrodes implanted in his or her head, and they really have nothing specific to do there, we have a unique chance of inviting those participants to perform specific cognitive tasks. And we have the unique opportunity of looking at very specific and circumscribed brain regions and to explore the specific modulations within those regions as they are engaging in different tasks. And this is just a very unique opportunity. And we have been able to leverage this approach for the first time in our understanding, to explore translation processes. So we had access to this patient, a young patient with an occipital-parietal alteration. She was a highly proficient bilingual with language skills highly spared. And we asked this person to engage in a translation task involving both translation from L1 to L2 and from L2 to L1. When I say translation, I'm talking about single word translation here. And what you can see here on the right is the location of the intracranial electrodes that were implanted in the patient, so that we have a fairly nice coverage of several regions that we know are relevant for linguistic processing at large and for translation processes more particularly. You can see that many of the electrodes were implanted in perisylvian regions, including inferior frontal and superior temporal structures, as well as some extrasylvian regions, which are also of major interest for both linguistic and translation processes. And so we focus on our analysis on two areas that are of very, very crucial for lexicosemantic processing. Within the posterior fusiform gyrus, we found that in a very early window, within 200 and 250 milliseconds after the to be translated word was presented, we found that forward translation from L1 into L2, involved a greater amplitude of the P2 component, which is a key index of attentional demands, than translating in the other direction. So very early on, this translation direction taxes attentional mechanisms to a significantly larger extent than the other translation direction. And another implication of this finding is how quickly the human brain can allocate specific cognitive resources that differentiate between translation directions. You may ask yourself, how long does the brain need to deploy direction-specific mechanisms? Well, as little as 200 milliseconds. That's an answer that could hardly be attained in the absence of these tools. And we have other results here, but I think that the main point of how you can have a temporally precise idea of the inner unfolding of translation processes, I hope I have covered that point. And let me emphasize this. There are many ways of tracking time in translation. You can do it by just measuring how long a person takes to translate the whole text, of course. Maybe half an hour versus 25 minutes if the person is experienced or non-experienced, or if they're doing it from their mother tongue to their L2 or vice versa. Or you can time how much it takes a person to start saying a word, the translation of a source item. Now, if you do that, what you're timing is actually the global time it took the person to do that. But what we are doing here is actually measuring the different temporal modulations that elapse between the moment in which the source text was presented and the moment in which the target text began to be produced. Everything here in the middle, between source text presentation and target text production, that's in particular what you can capture with these techniques. And you cannot do that with any other technique. You can also gain insights into how different mechanisms interact in the brain. And to this end, what you can do is look at how different brain systems are exchanging information, how they are synchronizing their activity patterns in time, or how they are desynchronizing their activity patterns in time. And there are different ways of looking at this. The main word here is connectivity. When I showed you the results from the fMRI studies, what you're looking is at how specific regions are increasing their metabolic demands and their activation levels during a task. But that is not telling you how any of those regions is actually synchronizing or coupling its activity with that of other regions. You can do that with connectivity measures, and there are different ways of different approaches to connectivity. You can take a look at structural connectivity using basically diffusion tensor imaging, or you can take a look at functional connectivity, in which basically what you do is you capture certain patterns of activity over a certain brain area or a certain brain topography, and you see whether those changes are occurring in a coupled manner with those in other regions or topographies. And we have used this approach to have a more widespread, multivariate, cross-regional perspective of translation in a few studies. This one, which we performed with Binghang Shen a couple of years ago, what we did was to assess different brain coupling patterns during sentence translation in Mandarin, Chinese, English bilinguals. And what we found basically is that translation from L1 into L2, that is to say what we call forward translation, involved greater activation exchanges, greater coupling between key areas that are involved in lexicosemantic processing, such as the anterior temporal lobe, with key areas that are crucially involved in attentional mechanisms, such as hubs of the frontoparietal network. And this greater integration of lexicosemantic and attentional demands was present in translation from L1 into L2, but it was differentially present in that direction. Because when you take a look at how different brain areas are coupling their activity in the other direction, when you translate from L2 into L1, that's what we call backward translation, you will see that such patterns are nowhere to be seen. What you have is basically a pattern of connection between the anterior temporal lobe, a critical hub for lexicosemantic processing, with the thalamus, which is mainly a sensory relay area that mediates processes that do not involve markedly overt attentional demands. So this is one way, and it's indirect, and of course it's imperfect, but it's one way to capture the way in which, in this case, linguistic and cognitive control mechanisms may come together differentially for one type of translation activity vis-a-vis another one. You can also use neuroscientific techniques to find correlates of competence-related and expertise-related effects in translation and interpreting. And mainly the field has focused on interpreting here. There are, fortunately, a growing number of studies have been produced that have explored how the development of interpreting competence, and sometimes leading all the way up to expertise, may involve different structural or functional changes in the brain, either the brain in itself or how the brain allocates different resources to actual linguistic and non-linguistic tasks. And we have used this approach ourselves in this study that we published in Neuroimage, a couple of years ago, I think, or last year. And what we found here is that if you compare professional simultaneous interpreters with non-interpreter bilinguals, that is highly proficient bilinguals, but who have no training and no experience in interpreting whatsoever, the interpreters, the professional interpreters, they are characterized by higher power along the delta theta band across many different linguistic tasks, including not just backward translation and forward translation, but even in single language tasks, such as L1 word reading and L2 word reading. And what is interesting also is that in the brain of the professional interpreters, and only in their case, the level of power along these clusters in the delta theta band correlates positively with translation speed. The greater the power is in these frequency bands in the professional interpreters, the faster they translate. And that correlation is not observed in the non-professionals, in the, let's call them lay bilinguals. To those, in those participants, the power observed in these frequency bands in the EEG signal is not significantly correlated with how fast they translate. So this is very interesting in the sense that this allows you to find direct correlations between outward performance in the translation task and specific neural correlates. So, bridging the two worlds, the world of what we can outwardly measure through behavioral indices with neural traces, I think that's one of the main directions in which the field should proceed, so that we can finally overcome the existing divide between the mainstream models of bilingual memory in the field of bilingualism, which have been mainly informed by strictly behavioral studies, such as the revised hierarchical model, or the bilingual interactive activation plus model, or more recently, the computational multi-link model, and the other strand of research, which is neurocognitively oriented. So I think that we can find a way between those two traditions, and even focusing on translation processes, and while studying forms of expertise in interlingual reformulation, as in this case. And of course, all these results, they can be used to directly test hypotheses and to formulate full-fledged models of translation and interpreting from a neurocognitive perspective. And this has been done not too often, I want to say. There are wonderful pioneering efforts by Michel Paradis in the 80s and the 90s. There's, to this day, a very, very interesting neuroanatomical models of the systems that intervene in simultaneous interpreting by Franco Fabro, published in 1999 in a very interesting book that he released at that point, and a few other models out there. But the fact is that you can use all this evidence not only to test hypotheses that emerge from neurocognitive models, you can use this evidence to test hypotheses that have been produced in non-neural trends in the field. And that's another point of synergy that I believe should be more exploited in years to come. And, more generally, all these methods, they open new ground. They lay the foundations for a new interdisciplinary avenue for translation and interpreting studies. Historically, translation and interpreting studies have, they have really imported many insights from many different fields. And this is a figure from a very nice book by Franz Pochak in 2004. And, insights have been derived from so many fields, ranging from anthropological studies, socio-professional studies, institutional studies, interactional studies, textual and cognitive. And there is no way why we shouldn't also benefit the field by incorporating neural evidence. And of course, it would be absolutely foolish for one to say that neuroscientific evidence is better or more preferable or more critical than any other source of evidence that's presented in this graph. But the opposite would also be equally false. To believe that there is any line of insight which is not useful enough or not as relevant as others is a form of reductionism that we do not want to embrace, I'm sure. So if we adopt this sort of equal opportunity employer perspective, for the way in which translation and interpreting studies has to approach its relation with other disciplines. The incorporation of neuroscientific insight is just a natural extension of its multi and interdisciplinary ethos, an ethos that has characterized the development of the field since its very inception, I should say. So just a few ideas to close up here. I do want to bring up something that has been known as the seductive allure of cognitive neuroscience. Neuroscience is full of problems, as any other field. The seductive allure is a bias that has been detected in the literature, whereby the very presence of neuroimages, of brain images, can raise the people's and readers' conviction and trust in the results that are being presented, even when the images that are being shown have very little to do with the actual information being described in the paper. That's one problem, and we really have to be very critical of the limitations of the approach, so that we don't end up endorsing wrong views and capitalizing on biases that may lead people to overestimate the relevance and the import of neuroscientific findings. And then again, another problem is the replicability crisis. Neuroscience is a field that seems to treasure novelty over replicability in a very uneven fashion. The main journals in the field, they will be much more, they will respond more favorably to a study that's presenting something new, novel, catchy, something that has somewhat of a wow factor, rather than publish studies that replicate previous reports in the literature. And that's a problem, because the very philosophy of the field pushes us researchers to be trying to do something new. And what you have as years go by is isolated reports of findings that do not really lead to repetitive testing and retesting under different conditions by different teams. And in fact, when specific consortia come together and different centers do make the deliberate effort to replicate widely cited studies in cognitive science or in social science, only a small proportion of them show the same results. Many of them do not. So it is very important, I believe, to make a distinction between an empirical result and a fact. By reporting a significant result in a very carefully controlled study in a well-reputed journal does not mean that that is a fact. And we really have to be aware of this and come to terms with the tentative nature of scientific findings, and just be careful in the level of assertiveness that we use when we are describing something that was reported in one or two papers, and not to confuse that with a factual and definite demonstration. That being said, the imperfections of cognitive neuroscience do not annul its contributions. After I say all this, you would say, well, I mean, so if neuroscience has all these problems, I mean, why even worry about it? Well, the thing is that every other field has its major problems. I mean, this is true of linguistics, of social science, of philosophy, of whatever field you can think of. And it is the point is not endorsing the false belief that a discipline needs to be perfect in order to be informative. What we have to be is aware of the limitations of each field, so that we can be aware of the scope of the interpretations that we can derive from the findings, and so that we can also be aware of the gaps that are present there, so that we can tackle them in successive studies. But neuroscience is not perfect, and the point is that those imperfections are not enough for us to skew it, to reject it from the overall panorama of lines that can inform translation and interpreting. If we demanded perfection from science, then science would not exist, basically. And like I said before, brain-based insights are not better than textual or cognitive or sociological insights, but the opposite is equally false. And sometimes it has happened to me that when you say, oh, so this process is observed by this region, some people may believe that that you are like finding an actual demonstration of something. That would be wrong. I mean, it's or that finally we are learning something concrete about translation that other approaches are not showing. No, that is absolutely wrong. Translation is a multi-layered, multifaceted phenomenon, and certainly learning about the sociological, the scopus-driven aspects of it, the ideological aspects of it, the institutional aspects of it is absolutely fundamental, and it is every bit as important as learning about the human brain. But to believe that any of those lines of insights are more critical than neurological insights would also be a mistake. So again, what I argue for is a balanced view in which we recognize that every possible strand that we can lean on to learn about what we are interested in is of value. And by way of epistemological notes, there are a couple of things I want to say. Now, there's something that I like to refer to as the misinterpreted copula and the reductionist fallacy. Let's say, and I probably said things like this during this talk today, this region is, or let's say maybe, if I say, for example, translation is a neurocognitive phenomenon, yeah? So something that may be easily said, oh, that's a very reductionist point. How can you say that translation is a neurocognitive phenomenon? Don't you think it's a scopus-driven task? Don't you think it's also a textual activity? Don't you believe that it's also a form of social interaction? And the truth is that when you say that A is X, you are not implying that A is only X. That is the misinterpreted copula. You can perfectly say that translation is a neurocognitive phenomenon, and that does not mean that you are denying the many other things that translation can be. You can say that it is a neurocognitive phenomenon, and at the same time, you can perfectly endorse the view that it is an ideological activity, that it has a textual dimension, that it is an institutionally shaped way of communication. And I'm saying this because I've come against arguments like this. And when you define translation as a collection of neurocognitive processes, denunciations are entertained of you being sort of neurobiologically reductionistic. Okay, so when somebody defines translation as a textual activity, should we assume that they believe that the brain has nothing to do with it, and we should denounce them as being as engaging in a textual reductionism? No. What we have to understand is that you can say that Adolfo is a researcher, and that doesn't mean that he's not also a father and a lover of metal music. You can say that water is transparent, and by saying that, you're not implying that it is not a liquid. By the same token, you can perfectly say that translation is a neurocognitive phenomenon and not imply that it is not any of the other things it certainly is. Another thing that I want to say is that it would be very easy and self-indulgent to say that, oh, now we are in the presence of a new shift for translation studies, the neurocognitive shift or the neurocognitive turn. And these are things that have been said so often in the field. Every 5 or 10 years, there's a new shift. The textual shift, the sociological shift, the whatever you want shift. And the truth is that all those, in general, all those so-proclaimed shifts have rarely amounted to more than new lines of study, which may be very, very interesting, but they have not really redefined and reshaped the field as the term may imply. And I want to be very explicit about this. I do not believe that we are in the presence of any shift or revolution or turn. If anything, what we have here is a line of continuity. Well, by incorporating neuroscientific tools, we're not recreating or reinventing anything. We're just adding new layers of insight to a tradition that has been growing up in a very pluralistic manner for decades and decades in translation and interpreting studies. And I really see no point in endorsing these catchy metaphors that are usually proposed for the field in attempts to reinvent something that, to be honest, is just a very virtuous, accumulative process of synergistic insights. So I want to be very clear about this. I do not believe that we are in the presence of a new stage or a dramatic change in direction. What we have here is just a new strand to build upon a very pluralistic line that has been guiding the development of the field for many decades, and that's what we have: continuity, basically. And of course, everything that I presented here and everything that you read in a scientific paper should be better presented as a finding, not as a truth. There's a good chance that many, perhaps all of the things that I presented today, the actual results, will be falsified as we move forward. And that may be perceived as a weakness, but I don't think it is. It's actually a symptom of good science. Science is not the space for ultimate truths and demonstrations. What we want to have is results that are consistent and that emerge from carefully constructed designs, experiments, and studies. And what you can obtain from that is a finding, not a truth. Experiments will, by necessity, be imperfect and partial because we need to control things, because we need to reduce the complexity of certain phenomena in order to make them manageable in a laboratory setting. And also, it will be flawed because experiments are constructed by human beings who are, by nature, definition, and necessity, flawed. So it is very critical for me to dispel any notion that anything that has been presented here may actually merit the label of a truth. And it will be actually very, very good if in a few years, I myself or any of you or anybody came and presented evidence that falsifies some or all of the findings that I found here. Because if that happens, it means that we are moving forward and that whatever was presented here was sufficient inspiration or motivation for better research and knowledge to emerge. And if we find compatible evidence, fantastic. Then we are accumulating results in the same direction. And that is also, of course, very, very healthy in its own right. So I have been, a couple of years ago, I have tried to consolidate all of these insights in this book. And in particular, in chapter 2, I believe, which is one of the longest chapters in the book, there's a detailed description of all these methods and many others. So for those of you who are interested in delving deeper into the nitty-gritty of the methods that we just very quickly went over today, please feel free to check out that volume, or at least that chapter, in which I gave it my best shot at consolidating all this information and many, many other aspects, ranging from the types of variable, the types of experimental design, the types of tasks that one can use to learn more about the neurocognitive basis of translation and interpreting. So thank you very much for your attention.
Well, thank you so much, Ardolfo, Dr. Garcia, for taking us with you on this fascinating ride and giving us with such a rich and systematic account of how the translators or interpreters' brain can be studied from what people cannot do and what people can do. And also telling us, how tools that are either sensitive to the regions or sensitive to the temporal process, of the translation and interpreting task can be used. And I also appreciate a very important note about the promises and the limitations of the approaches, especially the issue of replicability. And also, I truly appreciate, that very important message that you're trying to send us about this having this very balanced view. And I truly hope that all my students can hear that part. And finally, I absolutely love, what you said about there are no such things as turns or shifts. We should not emphasize too much about having all these different terms or shifts. And I think this is a very important message to send to people in our field, in translation studies and interpreting studies. Okay, we're going to entertain your questions now. And when we ask you to register, we did ask you to also send your question, and some of you did. So we probably will go over those questions. And of course, we want you, among our participants, to ask your questions too. The way to do it is by raising your hand. We'd love to hear you ask your questions yourself. So raise your hand and I will call out your name, and then you can unmute yourself before you ask your question. And of course, you can also use the chat function, to type in your question. Okay, so do we have the first question here?
Do you mean from the ones we had that were sent in advance, meanwhile, or should we start with the ones from the audience now?
I think we can start with the audience now, because this is how I see it. Because before people have a chance to listen to your talk, they might not be able to anticipate, what is covered in your talk. So I wonder, whether those questions, people who ask those questions still want to ask those questions. So I would like to ask our audience, our participants. I'm sure, those quick people actually posted that the questions are among the participants. Ask your questions first, and then if we have more time, we can entertain the other questions.
I think Judith Brenner is raising her hand.
Okay. Okay, so please, we can ask our staff. Yes, okay, go ahead.
Hello, thank you, Adolfo, for this fascinating talk. I am interested in the detail about the IEEG method you mentioned. You said that the patients who undergo this procedure are epilepsy patients. And I would like to know, how can you know that the findings from this method, because your group you're studying is only a specific type, how can you transfer these results to a broader group of translators and interpreters?
Thank you, Judith. That's an excellent question. And there are a couple of things I'd like to say. The first one is, we're working with patients with epilepsy. So the first question that may be raised is, okay, so you're looking at a dysfunctional brain. Is that a good approximation of how a healthy brain works, right? That would be perhaps one aspect of your question. And yes, of course, when you're working with patients, you always have this issue to take into account. In the case of our protocol with epileptic patients, what we make sure is that the electrodes whose signals we pay attention to, they have to meet a certain number of requisites. First, they have to have a clear contact point in an area of interest. Second, that area of interest needs to be sufficiently far from the epileptic focus, so that we can know that it is not affected by the disruption that typifies specific brain regions in, let's say, temporal lobe epilepsy or frontal lobe epilepsy. Also, we want those electrodes to be away from white matter, basically because then regional interpretations of the activity patterns are not that clear. And then there are a number of statistical measures that we take to make sure that the signals that we are recording are actually representative of the phenomena of interest. So there's that part of the question. Then the second one is, so you're working here with an epileptic patient, and this person in particular, she is. I'm going back to the time when we did the study. A highly proficient bilingual. She was actually an English teacher, but she was not a translator proper or an interpreter. So, there's always that, especially in the light of the studies that do show that neurocognitive patterns can differ between professionals and non-professionals. So the question is, can you really extrapolate what you're seeing in this person onto the brain of an actual interpreter or translator? And I would say the following: the fact that in comparing professionals with non-professionals reveals certain neurocognitive differences does not mean that there are no points of commonality in the very, very basic aspects of translation and interpreting between non-interpreters, or let's say non-professionals and professionals. Indeed, there are a number of mechanisms which will remain part and parcel of what translation and interpreting involve, irrespective of competence and expertise. And there's one more thing that I'd like to add. So, by the way, in that study, we were looking at very basic mechanisms of word translation. And then the second thing is that with that particular technique, you're looking at information that comes directly from the neural regions, from inside the neural regions that you're interested in. This means that those activity patterns that you're recording there, they are not so much affected by modulations in other neural systems that may be more susceptible to expertise or competence related effects. So those are mainly the grounds by which you can extrapolate the findings. And then there's another thing that is very important, which is that it is one thing for you to describe the activity of translation, and it's another thing to characterize how translators go about their activity. So it's different levels of resolution, of granularity, if you will. It's different constructs, and it is perfectly feasible for one to try to characterize what happens in the translating in the brain when it is translating, without really making a commitment to which specific population that may apply to. And that's basically the way in which we are framing these studies. We are not ready to make a commitment saying, okay, so this is how this will specifically work in the brain of someone who's a beginner translator or an experienced translator. But how you define your construct is important to see the extent to which your results may or not be or may not be generalizable. So here, I think it's better to think about this as information that may inform how a specific process in the bilingual brain occurs, in particular when you are engaged in this specific form of intralingual reformulation. Whether that is applicable across the board, across different populations, is something that we can, by default, assume is the case or not, depending on your theoretical inclinations. But it's something that, first and foremost, I think should not be should not be taken as a consequence of the data, basically. I would be agnostic about that, Judith, about that aspect of the study, and just focus on the things that the study can actually inform, which is this specific difference in the timing that and the specific modulations that certain brain regions exhibit when a bilingual person is engaging in this task versus this task. So I will be careful about extrapolating that, knowing that it could be done, but only if you assume certain theoretical or empirical commitments.
Thank you. We do have a couple questions typed in the chat box. One of the questions is, are there any good ways to measure EEG or functional MRI during target language production in interpreting, such that you can minimize the artifacts of movement?
Excellent question. So for the benefit of those who may not be familiar with why this is an issue, EEG and fMRI, they're capturing activity that's distributed across your brain. And when you move, when you blink an eye, when you tense your arm, when you move your tongue, when you move your jaw, all of that will involve major activity patterns in your motor cortex, basically. And that can introduce a lot of noise in the signal when what you are actually interested in studying is a cognitive process. So if you go over some of the papers that have looked at translation or interpreting, let's say here, as in this question, in fMRI in particular, you will, and in EEG studies too. I'm going back to the original work by Kurtz, for example, with Edmund Petsche in the 80s and 90s. And you will see that some studies, what they do is they opt to do something that's known as silent interpreting. So basically what they will ask is for the interpreters to perform the interpretation in their heads without opening their mouths, as if they were listening to their inner voice during the interpretation. So when you come across a paper that does this, you would say, why are they even doing that? I mean, that's not how interpreting works in the real world. Well, the reason why that's done is because it's like the most straightforward way for you to avoid or actually minimize the motor artifacts that would be involved if you actually have people overtly translate or interpret, which is a motorically loaded activity. So one approach is this, is you use silent interpreting or silent translation. What's good about that? You remove all the motor artifact, or most of it. What's bad about it? You cannot judge quality. You cannot judge performance. You cannot know whether what that person was interpreting in his or her head was actually a good rendition of the source text, how many errors were being made. To be honest, you don't even know if the person was complying with the task. You don't. So there's a great deal of uncertainty that comes with that. That's why some people opt to deal with all the motor artifacts, really have the persons engage in all the buccal-facial activity that overt production entails, and then deal with that. How can you deal with that? There are some statistical ways of doing this. First, you want to make sure that the head is as stable as possible in the scanner. In the case of fMRI, there are movement and rotation parameters. We usually account for 6 of them, so you don't want to have any rotation higher than 90 degrees and things like that, and you can account for that statistically. That's just like a very basic thing. It doesn't really account for the actual articulatory movements that we are concerned with here. One thing that you can do is to estimate, perhaps by the nature of the phonemic and phonetic properties of source text, of the target text that's being produced, which can be done post hoc, to show that the proportion of plosive consonants and fricative consonants and voiced and voiceless sounds and vowels, etc., that did not significantly differ between one condition and the other. If you can do that, you have sort of a descriptive proxy of the actual articulatory demands that were put in play in one condition versus the other. And you can actually show that this is not statistically significant, or you can enter those variables as regressors or covariables in your model, and you can see whether they really account for the variance that you observed in the comparison between the two conditions. Another thing that you can do is you can actually record the production and then use automated speech analysis to show and perform comparisons of articulatory features such as fundamental frequency modulations or the transition between voice and voiceless segments, or you can perform prosodic calculations and again show that those did not significantly differ between your two conditions of interest. So and one more thing you can do is you can have sort of a control condition which is specifically designed to account for motoric properties of the participants. And then you can use the results from those tasks as a sort of baseline for subtraction, or again, as a covariable. So I think that these are the main approaches I can think of. None of them is perfect, and all of them are tedious and costly in terms of time. But the summary of my question would be, you can avoid the issue by engaging in inner interpreting or inner production, whatever it might be, at the expense of having any way of judging the actual quality of the performance, or even assuring task compliance. If not, you have to deal with artifacts. How can you do that? You can do it by taking the phonological and phonetic properties of the transcribed productions as a proxy of articulatory demands. You can do it by recording the participants' output and engaging in statistical comparisons of relevant acoustic measures, or you can do it by including some sort of articulatory baseline comparison and then subtracting it from your conditions of interest.
Our next question is, so what can we truly expect now from neuroscience approach to enhance translation process? So this is more about the applications of the approach.
Thanks for this question. It's a broad question. And so what can we truly expect from neuroscience approach to enhance translation processes? All right. Let's see. First of all, a scientific field does not need to have immediate, obvious practical applications for it to be justified. That's the very first thing I will say. There is a tremendous value in the very adventure of discovery. And in fact, many of the translational or applicable developments in many fields, they were actually produced several years after a certain basic, more sort of a declarative discovery has been produced. So that's the first thing that I want to say, that irrespective of how I build up the second part of my answer, the justification and the contribution of the field in the broad sphere of science does not hinge on its having direct, immediate practical applications. That being said, I do believe that there are practical applications that can be derived from this. For example, we are learning that L1, L2 translation versus L2, L1 translation, but typically, I mean, at least in the Western world, it's more typical for people to engage in L2 and L1 translation than the other way around. This is a very cultural-driven dichotomy, I should say. In translation training programs, there's usually, I guess this changes from country to country, and I don't claim to know what's going on everywhere, but some of the programs that I know, they do emphasize, they do allocate more time to working on L2, L1 translation than the other way around. And then you may think, okay, so the reason why the other direction proves more costly might be because we do not devote so much time to it in our training. But when you take a look at what happens in people who have not received any training in any direction of translation, you will see that still one of those directions, typically in Western participants, L1, L2 translation is more effortful. It is more costly. And something that we are learning from neurocognitive research is what is driving that cost. It's not just the linguistic demands of the task. It's not just the cognitive control demands of the task. It seems to be the integration of those mechanisms. And to the extent that one becomes aware of the type of cognitive resources that are being recruited by a certain task, one can design better tailored activities in the daily or weekly training of practitioners, for example. Another thing that we that we can learn from here is specifically in the studies that compare experts versus non-experts, or people with higher levels of competence with those of lower levels of competence. And something that you will see is that the fact that you undergo a translation training process or an interpreting training process is not going to boost your linguistic skills at large. Something that happens is that so you will become better at the task of interpreting at large if you train interpreting. Let's hope everybody, all the trainers do their job right, and you have the minimal levels of aptitude and commitment to the task. You will become a better interpreter as a whole. But also you will be strengthening specific subcomponents, cognitive sub skills that are involved in the interpreting task. There's a good chance that your working memory will become better, if we want to believe what the literature is pointing at. There's a good chance that your speed for finding translation correspondences will be lower. Yeah, it will be faster. It will be higher. Sorry, it will be faster. And there's a good chance that you will be able to find suitable words for the task at hand in a more efficient manner. Now, there are other linguistic skills that will not benefit from your having undergone that training. From what we know, for example, your word reading skills would not benefit from that at all. There's no clear evidence that your task switching, that your cognitive flexibility will become any better. The evidence is very ambiguous about that point. There are other executive and linguistic functions that do not seem to benefit clearly from this. So it's also good to take a look at this evidence to have a clear idea of what the cognitive implications of a translation or interpreting training process may be and what may not be, so that we can create realistic expectations and so that we can create better, more specific tasks and exercises for trainees. So I guess those are the main things that I would have to say to that question.
Thank you, and thank you for emphasizing the nature of scientific discovery. The serendipity of great discoveries all the time, most of the time. Our next question is about whether this kind of research can always need to be done in a laboratory.
Thank you for that question. Well, if you want to produce evidence firsthand, yes, I would say there's not much way around that, but I will say two things. we are witnesses an emergence of neuroscientific tools that can be used in daily life settings. For example, there are portable EEG devices that can be used now. And that's actually very interesting if you want to have a more ecologically valid, naturalistic approach to whatever cognitive process you're interested in. Just as I'm saying this, I will also say that those settings that are meant to be used outside laboratory, they have all sorts of complications in terms of calibration, data quality, consistency, the very controls that you need in a typical experimental setting. So it's complicated. Another way in which you can engage in very useful and creative neuroscientific research on translation without entering the lab is by conducting and providing good systematic reviews of the evidence and using that information to come up with hypotheses or theoretical positions or models that do use and sort of build on the evidence that has been produced in the lab without you actually needing to go into the lab. Perhaps one of the best examples of this is one of the pioneers of the field, a person that I have the utmost admiration for, who is Michel Paradis. Michel Paradis was a founding figure in the neurolinguistics of bilingualism, and to my knowledge, he never conducted a single experiment. And it's only in a very few cases that he had direct contact with the patients that he reported. But he was an amazing, like a really brilliant consumer, organizer, and synthesizer of the literature. The work that, in my appraisal, had the most impact from the things that he did was to collect massive amounts of evidence out there and to see them in an integrated fashion and to find patterns that seem to align across different studies and things that were very inconsistent, and to come up with very reasonable theoretical explanations for those discrepancies. So I guess there is a way to engage, to work in this line of work without actually being within a laboratory setting. And these are the main things that I can think of. But if you really want to produce first-hand data and go and address your specific hypotheses, there's no real way out of getting your hands dirty in the beautiful mud of the lab.
Possibilities and also limitations, also possibilities. The next question is whether the emotional state of the participants evolved in these studies using these methods were taken into account, given the links between emotional and cognitive processes.
Fantastic question, and that's a limitation of the field. I would say that not a single one of the studies I showed controlled for emotional variables, and there's nothing to say in defense of that. It's a limitation. Sometimes, and it's a pity, because it's something that we typically do when we work with patients. There's a line of work that we have at my lab in which we explore linguistic markers of neurodegenerative diseases. And yes, we're focused on language, but we do try to collect data regarding depression, anxiety levels, overall the overall affective state of the participants, because, as this question very well highlights, that does have a direct impact on cognitive process and neurocognitive mechanisms at large. To my knowledge, that has not been integrated in the studies that I showed. That is not to say that emotional and affective aspects have not been studied in the context of translation and interpreting studies. There's a growing, a thriving line of research on how emotional and affective aspects impact translation. But to my understanding, these have these studies have mainly relied on either impressionistic sort of qualitative descriptions of participants. Second, on peripheral measures such as heartbeat or heart rate and heart rate variability, or measures of respiration, and sometimes even skin conductance response. These are all peripheral measures, right? All dependent on the autonomous nervous system rather than the central nervous system. So we know a lot about the neurolinguistics of translation and interpreting, and we are learning a lot about emotional and affective aspects of translation and interpreting. Those two worlds have not coalesced yet. So, and there's a chance that many of the results that I showed here, they may be confounded by different emotional and stress-related aspects. In fact, something that we know is that when you compare stress levels, for example, via peripheral markers in experts and non-experts, for example, during interpreting sessions, the professionals are much better able to regulate the markers of stress, for example, heart rate. And to the extent that heart rate activity does modulate a number of neurocognitive mechanisms, let alone because of the impact that the heart evoked potential may have through the vagus nerve on different neural mechanisms, there's a chance that when you compare a professional with a non-professional and you fail to account for emotional aspects, you may be missing out on one of the domains that accounts for a good proportion of the variance. So that's a very good point. It is a limitation, and it will be very good for these two fields to come together, to join hands, so that we can start factoring in emotional aspects in neurocognitive research on translation and interpretation.
Adolfo, I'm very mindful of your time and also our participants' time, of course. So we do have probably 4 questions. How do you want to answer them? Are we going to take a little bit more time to answer them, or?
We can take a few more minutes, and I'll try to be as brief as possible. That's okay.
Okay. The next question is actually a very broad question. Which universities or institutions allow for this type of research to be done, and whether how to have access to this to these institutions?
All right. So I mean, centers and institutions that are engaged, that partake in neurocognitive research, well, there are way too many to be counted. Now, institutions that have neurocognitive equipment and laboratories directly and specifically devoted to translation and interpreting, I can tell you about the ones that I am aware of. There is the TRACO lab, the Translation and Cognition lab at in Germersheim, under the direction of Dr. Sylvia Hansen-Schieder. And they have EEG and eye tracking devices, and they use those tools specifically to study translation and interpreting. There's another laboratory that has relevant tools, including FNIRS, that's functional near infrared spectroscopy, as well as eye tracking. And there's a chance that they may have incorporated new technology that I unfortunately may not be aware of, at Macau University in China. There's also at Durham University, Professor Bingham Shen has a laboratory that's devoted to studying interpreting and translation using neurocognitive tools, thanks to an alliance that he was able to set up with the brain science department at the university. Then in South America, we do it in our lab, of course, the Cognitive Neuroscience Center at Universidad de San Andrés in Argentina. And I should also mention the LETRA team, led by Dr. Fabio Alves in Belo Horizonte in Brazil. There is also the newly founded MC2 lab, led by Dr. Ricardo Munoz in Italy, and I guess I should also mention the Trieste, University of Trieste group, who were foundational in the use of experimental approaches to translation and interpreting. My sincere apologies if I am forgetting to mention any relevant lab. It's such a tricky thing to do, but I'd rather give specific responses and just make explicit that I may be forgetting someone and just give you a vague answer.
By the way, Professor Munoz was in among our participants. I don't know whether he's still here. And our next question is, in neuroscience or brain science, what is the relationship between translation and language production, that is, without this or source text? Is there any fundamental differences shown in the experiments? Yes, so this is the essence of the question.
Yes, that's okay. That's a good question. We know that there is a fundamental difference between translation. What I like to call intralingual reformulation. Let's say translation or interpreting or any variation thereof. Usually, what we do is we do a lot of site translation, for example. We know that there is a substantial difference between translation and single language processing. So when you show a participant a word or a sentence, and you ask that person to translate it into another language, and you compare brain activity patterns relative to when you ask that person to read that stimulus in the same language, there's all sorts of differences. The brain areas that become activated are different. Those that are shared between both tasks are typically more activated in translation than in the single language task. The electrophysiological signatures, ERPs, or oscillatory measures, they vary, they differ, and I think I showed some evidence in this direction. But the specifics of the question that was asked here was without a source text. So in the examples that I'm mentioning here, I'm comparing translate. Now, when you have a word, there's a stimulus, and you either translate it or you read it in the same language. But there's a source text. That's that source word or that source sentence in both tasks. I guess that what the question really aims for is when you have translation versus spontaneous production. When you ask a person to tell you, I don't know, maybe what a typical day of their lives is like, or to share a memory, or do something like that. And I have to say that I do not know of any study that has looked at that particular difference. We do know that there are substantial multidimensional differences between having stimuli and translating them versus just reading them or comprehending them in the same language. But a comparison in which stimulus-based translation task was compared with a non-stimulus driven, more of a spontaneous self-evocation production task, I don't think has been done so far.
All right. The next question is there new research in your area? I assume using the neuroscientific approach that is applied to remote interpreting.
I think that the answer is very brief here, and it's no, not that I am aware of, and it would be very interesting to do so. I am not aware of any study that has done that, but I can think of a couple of ways in which it could be done. So, for remote interpreting, you would require the participant to be facing perhaps the laptop with the Zoom interface, and then the interlocutors on the other side of the world, wherever it might be. And so what you could do perhaps is, I can imagine one way of looking at this with an EEG device. You would need the participant to be probably, sitting. So if you really want to recreate the scenario, So maybe with EEG, that would be one way of doing it. Another one would be to do it with FNIRS, with functional near-infrared spectroscopy. Both approaches would be so challenging in the sense that you would have so much artifact to deal with as the person is really engaging in the task. And then it will be interesting to think about what would be differentially learned by doing an interpreting experiment the way it's typically done, which is just presenting maybe a 4-minute recording, and have the persons, the participants interpret them as to record their brain activity, relative to having them engage in remote interpreting. If you're interested in exploring how the specific visual feedback coming from a computer impacts on it, maybe that's something that could be operationalized. And going back to the core of the answer, I don't think it has been done. I can envision a couple of ways in which it could be done, but it will be tremendously challenging. And to embrace those challenges, a very clear need to be made for why we want to do it that way, relative to more standard, less naturalistic, but more standard and more interpretable ways, I would say.
Okay, this will be our last question, and this is about, I think this is related to the previous question about emotions. So how about the nutrition and an exercise being factors on cognitive ability?
Well, nothing has. I personally do not know if any study has looked into this in the particular context of translation and interpreting. Now, in general, we know that both eating habits and exercise have a tremendous impact on cognitive ability. Exercise, sedentarism, or if you want to see it the other way, I mean, lack of exercise is one of the main predictors of cognitive decline in early aging. People who do not exercise regularly, they have a higher chance of developing dementia and other neurocognitive disorders. They have higher chances of developing mild cognitive impairment and even subjective cognitive complaints as they age. Eating habits are also fundamental. There's a lot of evidence coming now from a number of groups showing that the microbiota, which is tremendously susceptible to what you eat, when you eat it, how often you eat it, has direct impact on your cognitive outcomes. So absolutely, this is beyond what I can say given my training, but if we are to follow the advice of people who are experts in cognitive reserve and in healthy aging, exercise, balanced diet, healthy sleeping routines, and maintaining active social bonds as well as intellectual challenges are 5 of the main factors that contribute to cognitive reserve, and that proves all the more important as you age. It's controversial whether bilingualism is also a significant contributor in this sense. There's a lot of evidence that suggests that it is. There's a lot of evidence that suggests that it may not be so important, and we could have an entirely separate talk about that. But yes, definitely, exercise, sleeping habit, balanced diet, and maintaining active social bonds are fundamental to keeping a healthy brain.
Not directly related to translators and interpreters, but related to everybody. So this is a very, very good concluding answer for our session today. So thank you so much again, Ardolfo, for giving us so much to digest already we have participants asking whether there will be recordings available. And yes, the recording of today's lecture will be available on the website of the translation, of the Center for Translation. And I would also like to thank all the participants for your wonderful questions. So this will be the end of our session. And again, we hope that we will see a lot of you in our next activity. And before that, well, goodbye, everybody, and thank you very much. And thank you, Rodolfo.
Thank you all. Thank you very much. Looking forward to the next event.
Thank you. We're going to stay for probably 10 seconds and for the messages to come in, a lot of appreciations. All right, again, thank you and goodbye.